Drug target validation is a critical step in the process of drug discovery and development. It involves confirming that a specific molecule, usually a protein or nucleic acid, is a suitable and viable target for the development of a therapeutic intervention, such as a small molecule drug or a biologic. The goal of drug target validation is to demonstrate that a particular target is directly involved in the disease process and that modulation of its activity can lead to a therapeutic effect. This is typically done through a series of experiments and analyses to gather evidence supporting the target's role in the disease, as well as its feasibility as a drug target. The following are some key steps involved in drug target validation. Biological relevance. The first step in target validation is to establish the biological relevance of the target in the context of the disease. This may involve studying the target's function, expression, localization, and regulation in normal and disease states. Techniques such as gene knockout or knockdown, gene expression profiling, and immunohistochemistry may be used to determine the target's role in disease pathogenesis. Genetic and epidemiological evidence. Genetic and epidemiological studies can provide important evidence supporting a target's role in disease. For example, genetic association studies can identify genetic variants that are associated with the disease and map to the target gene. Epidemiological studies can establish a correlation between the target's activity and disease prevalence or severity. In vitro and in vivo studies. In vitro experiments using cell lines or primary cells can help assess the functional role of the target in disease pathways. In vivo studies using animal models can further validate the target's relevance and assess its druggability. Animal models may be used to demonstrate the target's involvement in the disease process, assess its tissue distribution, pharmacokinetics, and toxicology, and evaluate the efficacy of interventions that modulate the target's activity. Target specificity. It is important to ensure that the target is specific to the disease of interest and does not have significant activity in normal physiological processes. This can be assessed by examining the target's expression in normal tissues, studying its homologs or related proteins, and evaluating its potential off-target effects. Feasibility as a drug target. The target's druggability, or its potential to be effectively modulated by a drug, is another important aspect of validation. Factors such as the target structure, function, and accessibility, as well as the availability of suitable drug discovery tools and technologies, can impact its feasibility as a drug target. Reproducibility and robustness. It is crucial to ensure that the results supporting the target's validation are reproducible and robust. Multiple experiments and studies, conducted by different researchers or laboratories, should be able to confirm the target's role in the disease consistently. Clinical relevance. Finally, the clinical relevance of the target should be considered, including factors such as the disease prevalence, unmet medical need, and potential patient stratification or biomarker strategies. In conclusion, drug target validation is a critical step in the drug discovery process to confirm the suitability and viability of a target for therapeutic intervention. It requires a multidisciplinary approach involving biological, genetic, epidemiological, in vitro and in vivo studies, and careful consideration of the target specificity, druggability, reproducibility, and clinical relevance. Successful validation of a drug target provides the foundation for further drug development efforts and ultimately the potential for new therapies for patients with unmet medical needs.